Kind of. Okay, number one. Should I go over this one at all? It's talking about cytochrome C, similarity in proteins. Cytochrome C is a good example to use. It's not the only example, but probably on the AP exam, that's the example they will gravitate towards. Cytochrome C is just like a piece of like metabolism. It's an enzyme of metabolism. So since metabolism is conserved, it should be like highly conserved between creatures. The differences in the sequence of amino acids between cytochrome C and different creatures would give you like a logical explanation of who's related to who. But we're okay with one? Okay, how about two? Yay. Wow, thank you. Are we equal with two? Okay, well how about three? Countercurrent exchange. We talked about that like as in the gills of a fish. That allows them to do what? Pick up oxygen a little bit better, I think, is what it was. It was like a modification of their respiratory system. So 3 is okay. How about 4? 4's cool. What are we talking about here? We're talking about light reactions producing two things. What are the two things that light reactions produce? NADPH and ATP, correct? Now, a good question, since we're talking about that, a good question that I saw or that I had from a lot of you the other day was the question from your lab packet where you took photosystem 2, which occurs first, and photosystem 1, and recognizing that as your electrons are passed from photosystem 1 through this electron transport chain right through the middle here, and bounced up, there was a 50% chance of them going back over here, and a 50% chance of them going forward over to the reductase enzyme, which is way over here. Correct? What else was happening right through this step? Something else was coming in. Hydrogens were coming in. And these H pluses are acidic, correct? So as the H pluses come in, you relieve that pressure by going through what? They go through ATP synthase and they generate ATP, correct? So this idea of a cyclic flow, which is what we see right there, is going to allow for an increase in ATP production, right? Cool. Yeah, that's a really fun question. Anyway, the answer to 4 definitely was A, but I like going over cyclic electron flow a little bit because it was a topic that I saw like heavily discussed the other day. How about question 5? Are we cool with that being a picture of crossing over? What's another like key term that you associate with the word crossing over? It makes a certain type of chromosomes, right? Which is a nice way of saying that your chromosomes are no longer the original copies that you remember them as being. They're modified to some degree. Number 6. It says ATP is produced in all the following except. Okay, this is a good question. The answer, of course, is going to be C, the Calvin cycle, because the Calvin cycle, of course, uses ATP. But glycolysis, it produces ATP. By what mechanism? Substrate or oxidative? Perfect. How about the Krebs cycle? Substrate. How about cyclic versus non-cyclic? That's just going to be normal ATP production. You really can't like put it with anything else. The electrons transport within chemiosmosis. Yeah, it's definitely ATP synthase, which is going to be oxidative. But the Calvin cycle uses ATP. Another good question that they kind of try to tackle with that same like logic that's right through there is knowing that ATP is produced in both respiration and photosynthesis. Because you learned like in the 9th grade that it was like ATP mitochondria and it's exclusive, and now I want you to see that it's not exclusive to mitochondria. It's everywhere. Number 7 is, again, that's a horrible question. A stipe, that's a blade of a seaweed. Root, definitely probably wasn't maybe a root. because a stem is like one type of a not even really a tissue. A stem is like one organ, and the root's a different organ. A frond, I believe a frond is like the compound leaf of a fern. I think that's what you call it. That's what a frond is. Which leaves you with prothallite and rhizomes. Prothallite, and I looked this up the other day, but I've forgotten, which tells you how important I guess my mind perceives it. Prothallite are like these little balls that are on ferns, and I forget what their purpose is. I think they might be the gametophyte of a fern. But definitely the answer is rhizome, which is a modified stem. But it looks like it's almost a root, and that's kind of what you're talking about here. How about question 8? We cool with 8? You want to talk about it? Okay. How about 9? Most directly for water's properties. I would probably put D, the hydrogen bonding. I'll get it.
Sorry.
10. Circular chromosomes is going to be what? Definitely bacterial. If you think about it, A, B, D, and E are all pointing to what? Definitely eukaryotes. Eukaryotes are going to have like what I would call linear chromosomes. How about 11? Active transport. Who's responsible for that? Or it says, which of the following components of the cell membrane? Definitely proteins. Now, what are your 2 major components of your cell membrane? Do what? Yeah, phospholipids and proteins both. Now, what are some other minor things, like some minor members, some minor ingredients? Cholesterol's 1. What else? Carbohydrates, what do they do? Communication, recognition. The cholesterol's fluidity. Okay. How about 12? We cool with how you would measure how evolutionary fit an organism is? By the number of babies it can produce. How about number 13? Definitely can't be C, right? Can't be the last phase of it, the last part of interphase. It's kind of an impossibility. It moves homologous chromosomes to opposite poles. Bless you. I would say no, that's going to be describing meiosis 1. I definitely don't think C could be it. It is also known as cytokinesis. Is that true? Cytokinesis is right after mitosis. That leaves you with B or D, I think. And it says it regulates the transfer of genetic information from one daughter cell to another. So what I'm getting from like D, and I think some people pick D, and D almost sounds good, but you have to really slow down. The way mitosis works is you have, or the cell cycle works, is you have the parent cell, the daughter cells. The parent cell gives the information to the daughter cells, much like this, correct? But I think D is trying to explain like how they transfer to each other, how like they're sending information. Do what? Oh God. But I think this question D is like trying to show you like how they exchange with each other. I just don't think that's possible, or maybe it's possible, but it's certainly not like what mitosis is defined as. How about 14? We'll go through that one maybe. It says, which of the following methods for transport of CO2 provides a buffering system for the blood? When you talk about CO2, and if you look at a cell, this is a cell right here, it's likely going to be releasing CO2 and H2O. And what do those combine to form? We see this in the root system already, but we're seeing it now in cells too. What do we produce? Carbonic acid, H2CO3, and what does that produce? Perfect. And that right there is a buffer that you would use or find in blood. Cool. Which of course is going to be like all about B. Now the reason bicarbonate is a buffer, off to the side, I'd like to show you this. Remind ourselves that if you have an acidic solution is going to be high in H plus, and if you have something that is basic, like I'm going to show you down here, a basic solution is going to have something that is low in H plus. Well, if you put this HCO3 minus in a acidic solution, a solution with tons of H plus, it's going to form H2CO3, which is an OK. Yeah, HCO3 minus. And if you put the HCO3 minus low H concentration, it's going to release H plus to form CO3 2 minus, which itself is an OK molecule. So there's really no problem either which way. Makes sense how that works out. Cool. Here. Send this. Thank you. It's for your like 120 test score or quiz score. No. Perfect. Let's look at 15. How about 15? Shall we talk about 15? 15? Okay. So it says the population of the country shown in the figure above. And we can see obviously like there's tons of young, because remember this is going to be like young, my bad, young, middle, and old. And it works out that like there's not a lot of older people typically. There's a medium-sized amount of middle people, and there should be like a fair amount of young people. And the way like our country would look is our country looks something like this. Let's look how we would look, which basically is we're pretty much stabilized. There's a little bit of a bulge right through here, but that's because of what? Do what? Yeah, baby boom. Post World War II. Right? But what are we seeing in this case? We see like a tremendous count of like people that are very, very young. So it says, what's going to happen to this population? Stabilize in 20 years. Well, first off, what's going to happen to these young within maybe 10 years? They're going to reproduce. And of course, what's going to happen? They're going to have tons of children, right? And so what I see over the course of time is I see something looking more like this over time. Like if you continue this trend and these guys move up, there's nowhere to go but this. You know, imagine this is a triangle. It would make logical sense. Now, the only thing I could say is that either that's going to happen or there's something that's preventing people from living at about beyond this point. Maybe for some reason, like, and this is like kind of sad to think of, but maybe like 80% of the people were to be killed off by this age, or the population, which is completely unlikely, but you know it could be the case. So looking at this and looking at this slope that's created, I would say it's more of a guarantee that they're going to increase for quite some time. I don't think you're going to decrease, and I don't think you're going to reach zero population growth at this rate that you have there. So I would say B is the best answer. They could stabilize, but then stabilization would be meaning that they have to do that. And it becomes unlikely that you do that. You know, even if you think of baby boomers looked like this. They weren't this extreme though. They did have a bulge like this, but even they evened out over time. But this is an extreme case of growth of the young. There's tons of young here. How about 16? So 16 is cool. Prezygotic barriers, which is an accept. If it says prezygotic, that means before you make the baby. And then this is an accept question, so they're looking for really postzygotic mechanisms. I'd say the best answer is C. Like a reduced hybrid fertility or reduced hybrid viability. Fertility meant that the intermediate baby can't make its own babies. How about 17? Definitely can't be A, right? So respiration, not it. Not replication, not germination. The only option is translocation, I think. And what do we normally see? Which way do sugars move? If you're talking about like a plant, yeah, typically your photosynthetic product moves this direction, correct? So you move to the roots. Is that always the case? When would be a time where you would actually go from roots to top? Yeah, post winter dormancy. It says all the following are included in the modern concept of biological species except which one? I'd say definitely D. Because it says gene flow inhibition, which is basically saying that your genetics don't match. And if your genetics don't match, that means you're kind of not supposed to have a baby. It says AIDS is caused by the retrovirus HIV. Which of the following makes DNA complementary to the viral RNA? So they're basically saying what takes your mRNA from the HIV genome and transencodes it or encodes it into a form of DNA so that you have HIV but of the type DNA. What's the name of that enzyme? Right. It's a guarantee that this question in some form or fashion will show up on your exam. It's likely that it will show up in the multiple free response portion too. Like they always try to go and get this topic, and I couldn't tell you why they go over it each year, but somehow this will be probably a part of an essay, I suspect. Which do cellular organelles and eukaryotes have both electron transport systems and chemiosmotic mechanisms? I kind of think that we've already predicted this question a little bit, but what's the answer? See, I see like a lot of connection between this question and really a little bit of number 4. You could have asked like 4 and 20 in about the same way. This idea that they want you to know that in your mitochondria and in your chloroplasts, you have ATP being produced. Because if you think about it, why would you use electron transport systems and chemiosmosis? Like to do what? Yeah, definitely it's going to be all about making ATP. To me, 20 and 4 are the same question. Plants' lateral roots. And it says the word arise, which means you got to be thinking about maybe something along the lines of embryonic tissue. Like almost a meristem, right? Now, to me, it would make sense that we not choose the xylem or the phloem because those are already tissues, and we're talking about new roots coming from, correct? What would endodermis and epidermis sound like? Like, to me, that's like going to be like right within or right deep to the like outside. Epidermis, though, is definitely outside, right? So I'm thinking of like an outer tissue. And the answer, I think we've learned over like the last week or so, is the pericycle. Remember that when you have a pericycle, you're looking at that zone of new growth, correct? And as the pericycle grows outside or to the outside, you have what's called lateral growth. Well, that lateral growth, is that going to be primary or secondary? Definitely secondary. What is primary growth in a plant? Upward. When they say the word meristem, what does that mean? Do it? It could be an area of growth, right? It's just maybe embryonic tissue. How about 22? Me? Got it. Say that again. So, an ion pump? What about an ion pump? Like a sodium potassium pump?
Okay, but H plus not count. It's still an ion.
Technically, those are not called ions as much as they're called proton pumps. But H plus is an ion, and so like it is confusing at that point because the H plus, like you would think that like H plus movement and ion movement would be one and the same. They're really not, because H plus movement is typically through like a special type of protein. Moves across the membrane. So right away, like whenever they stop with this point at membrane, I think of the word diffusion. Along a concentration gradient at a rate faster than would be expected by simple diffusion. So obviously it's not simple diffusion. What types of diffusions do we have? What types of movement? We have simple. Then the next like level up is going to be what? Facilitated. And then we could be a certain type of facilitator called osmosis, correct? And then lastly, we could be active. And I'm going like through this in what my mind is like the levels of strength, like the tier system that exists here. Simple's like no energy requiring, no protein requiring. It could happen anywhere. Active has got to be requiring of a protein and some energy to do it. So it's very specialized. Good. So, but it says without expenditure of energy. So right away, active has got to be out of the question here. It says the substance moves across, and they never really state that it's got to be only water. So I'm really iffy on if we should talk about osmosis as being that mechanism there. So definitely not active, definitely not an ion pump. What does penocytosis mean? It means like cell drinking, like it's a form of endocytosis. Phagocytosis is eating like a big particle. Penocytosis is just eating like the fluid or drinking the fluid that's directly around the area. Osmosis is moving to water, and I really don't like picking that answer right now because it never said it's got to be water only. They made it sound like something has to be facilitated. Just the fact that they said protein but no energy, got to be facilitated. How about 23? What would happen to an individual playing a long game of basketball outdoors on a hot afternoon? Obviously, they would start to sweat. Like that's, I think, the first thing that comes to someone's mind is like the sweat, which is obviously the loss of what? And why would you do that?
Cooling.
Evaporative cooling. So there's definitely going to be a loss of water, right? Now let's kind of start taking some of the answers and let's remove the ones that make zero sense. C, the volume of urine would be produced. Like you've lived long enough, you know, and like it's pretty obvious. Like I don't go outside and play basketball. I don't really like to go outside. You see from my like white legs, but nevertheless, if you were to go outside, would you? I mean, play basketball for hours. Do you think your urine output would increase? It probably would do what? Decrease because, right? You're trying to conserve water. You're sweating it all out. So I would say C could not be it. The concentration of urea in the urine would decrease. Well, to me, if you pick C, you'd have to pick D too. Because these two are going to go hand in hand. Right? I mean, you're losing water, so obviously the water's got to come from somewhere. Thyroxine, it says, would decrease. What does thyroxine do? Increase metabolism? Do you need more or less metabolism when you're playing basketball? More, right? So you need more energy. So I don't think your thyroid would be decreasing thyroxine output. Remember, thyroxine was the hormone that like allowed for your metabolism to basically pick up. So I don't like A. The osmotic pressure of the blood would decrease, or the secretion of ADH or antidiuretic hormone from pituitary gland would increase. I think you're left with like B or E. You know, we know E is for sure going to be the answer, and I would say rather than trying to disprove B, I might focus my efforts on like proving how E is definitely going to be the case. Because as you release ADH, what's that going to cause? Your kidneys are going to do what? Water retention. Cool. It says the synthesis of a protein or carbohydrate polymers always produces which of the following is a byproduct. The synthesis of protein or carbohydrate polymers. So when they say synthesis of protein and they say polymers, I'm thinking of taking like a little amino acid, another little amino acid, and I combine them together to make like what I would call a dipeptide. Two amino acids that are connected together. What type of reaction is this where you are making something from two little pieces?
Dehydration.
Perfect. Dehydration. Give me some more though. Is this going to be anabolic or catabolic? Is this going to be a reaction that's likely or not likely? So the delta G should be positive. So not spontaneous reaction. 25. This ends like our first column. How do we feel about 25? This one sucks. This percentage is like close to basically non-existent. Now, but let's kind of start removing some things that make little sense, and let's see, at least give ourselves a good chance. Vascular bundle. Seems weird. But when we say the word vascular implant, what has to go through your mind right away? Got to be the tissue of transport, right? The tissue of transport, what's its function? Obviously, right? Can't be about doing like C3 photosynthesis. So I think I would get rid of E. Now, we learned earlier that the pericycle was all about what? Like growth area, right? Lateral growth. So I think then D goes away. The epidermis, where was that at? And what is the outside is like the cuticle protection area, right? The strong cell wall. Are we thinking that's where we're talking about? I don't think so. Now, this would have worked out perfectly if they had asked you for the cell type, because you would have said what? Parenchyma. The metabolic like type of cell. But they wanted what? They wanted, I think, like a type of tissue almost, right? So, C3, which is going to be a little bit different than C4, I'd say the answer here is going to be A, the palisade mesophyll. And that's really just one you kind of have to rote memorize, I think.
The outer part of the mesophyll.
Yes, not the inner part, because the inside part would be for C4. I'm sorry. Yes, there's a cross section. I wish I don't have the option to show my laptop, but if you look at chapter, I could. I'll tell y'all what. We'll take a quick break. I need to stop this recording anyway. Okay. Number 26, it says early spring flowers growing in a deciduous forest are adapted for habitat in which of the following ways. So this question stinks a little bit because you really have to know like the concept of like how a deciduous forest works. Let's focus. Get it? I need to work on new material, I think. But tell me about a deciduous forest. I mean, like, we definitely know what a forest is. Tons of trees. The trees are tall, and they are like very thick green trees, right? Now, describe like these little flowers that would be growing like in the spring. Do you see them being like as tall as the trees for sure? No, they're going to be very short, right? So to me, you would have to be concerned about like the amount of light that they could get. Now, let's focus on maybe just the idea of it being early spring. Describe, and this is a great time to mention this because this was like all of about 4 weeks ago here. Describe like what early spring is for plants. You've got not many leaves on the trees, right? But you certainly do have like these little flowers that pop up. Now, over the course of time, what would happen to those little plants that you could see on the bottom? They'd go away because your leaves would come in from your plants and block the light. So I would say your best answer choice here is going to be B, taking advantage of sunlight before sunlight is a limitation. Now what they'll do is they're going to produce their little spores, and then they'll produce like their little, probably just their spores, probably their little zygotes, little seeds. The seeds will be like saved for the year. They'll resprout the next year. Which is why, like, if you have a yard that you're trying to keep up, you always want to use like what's called a pre-emergent, which is something that can kill the little zygotes before they spawn. 27. 27 is a really good question too. To me, basically, you can eliminate C, D, and E. I think that if you missed the question, you probably eliminated C, D, and E, and you mischose between A and B.
Yeah, I was guessing between A and B.
Yeah.
That's wrong.
And you.
It's after. That's the confusing part because it was after the stimulation. I was thinking it was like trying to get back to normal.
No, I would say like this isn't like back to a normal. This is going to be like back to like stimulation. Just means like that those ion channels are turned on in that area as you carry it from.
So the restoration is the stimulation.
Yeah.
Okay.
And we know that Na plus is going to be moving in and K plus is going to be moving out. And then once that happens, who's going to fix that issue? The pump, right? Which is going to use a facilitated method. How about 28? It says, which of the following is true of intracellular junctions, such as tight junctions, desmosomes, and gap junctions? First off, bye. Have a good time at your performance today. When do y'all perform in like 2 weeks, right?
Sounds good.
Can I get tickets to go?
May 5th.
Is it like can I go to it? Y'all do have to leave your test though, sorry.
Oh, really?
Yeah. But if you want to, we can start like a special stack for the people that were here so that it's easily found next time. You can just set them like right there. I'll keep track of them all, or try to. Good luck at your show though. Go Mustangs, thanks for being here. Yep. Have a good time. Tell your friends. Okay, tie junctions, desmosomes, and gap junctions. First off, when I see these, this is what I think of first. I don't even know if this is the answer because I really haven't read the rest of the question. But animal cells. What's it called in a plant cell when you have like the gap junction in a plant cell? Plasmodesmata. That is basically the junction in plant cells. Now, with that being said, I don't even know if this has addressed the question at all. That's just what goes through my head. It says they are specialized forms of communication among nerve cells. Certainly not A. Their primary importance and the contraction of skeletal muscle fibers. To some degree, some of these are, but not all of them. And it says they are the primary importance. The primary importance is going to be microtubules and the use of ATP. So I'm thinking that B could be right, but it's likely that B is going to be not right. It says they are equally distributed among all tissue types. The fact that's start to distribute that's start to make an always case, and I hate always answers. So I'm probably going to eliminate C, because I know that they're certainly not evenly distributed. That would be so hard to do. It says they help integrate cells into a functioning unit. Now D is the best response I've seen so far, so I might want to pick D. It says they aid in distributing DNA among cells. Not really. That's actually more of the responsibility of microtubules. So if you pick E, then you really have to pick B as well, I think, because they're kind of going about the same way. I like your shirt, Caleb. Cool. Cool. How about 29? It says a person with systolic blood pressure of 120 and diastolic 80 typically has a mean arterial blood pressure of 90 mmHg. Which of the following offers the best explanation for why it's not, or for why it's 90 mmHg and not the average between the two? So typically when you average two things, like if I was going to average an individual's grades with a 90 and an 80, I would add them up, right? Get 170 divided by 2 and get 85, correct? That's how you do normal averaging. But is that how you would do averaging like in my class necessarily? Like let's say your test average is like an 80. Well, I take that and multiply it by 0.5. Let's say your lab grade is 100 and multiply that by 0.2. Let's say your homework grade is 100, which is 0.1. And what's left? 0.2? 0.2 for quiz grades, which is 90. So that's actually how I do your grades. So what is that, 40? What's that, 20? 10? And what is that? 18? So 40 and 60, 70, 88 would be the individual's average. Why do I not just add 80, 100, 190, divide by 4, done? Weighted, right? Now, we see a lot of times that it's either a weighted percentage or it's a percentage based on time. And that's, I think, what 29 is kind of alluding to. You wouldn't average these two numbers because your diastole is going to last longer. It lasts a longer percentage of the time. Make sense? Now, that would make sense because, look, the average that they are giving you is like 90, correct? And you had 120 over 80 is the blood pressure. So you'd have to have something closer to the 80 than you do the 120. Right? Blood pressure, the arterial pressure is determined just like your average of your grades. 30 is kind of a unique question. It's not this difficult, it's just there's a lot of room for error. It says, what stage does gastrulation begin? The answer is E. Gastrulation is this event that you see right here. And it's actually just starting, it's not even done yet. The infolding of the cells to make all the tissue layers. I think most, and I would be in the same boat too, wanting to pick like not E, because like you've got like you kind of feel weird picking like the last step is where the event that they want you to like. I don't know if anyone else has that phobia, but I do too. But all they show you like in 1, 2, 3 is like, and even really 4 is cleavage. 4 is just showing you a picture, but when I say cleavage, I don't mean like Bradley Hume's type, like what he says in class, 4th grade students. I'm talking about like the repetitive rounds of mitosis. He just, every time I see cleavage for he can't control himself. Oh, he's funny. 31. Echinoderms and chordates. What feature? Does anyone know this just off the top of their head? Like it was just like such an important feature. Deuterostome? Which means what? The anus first. But yes, the butt's first. You know, you can make little jokes about how to remember that. I don't know. But somehow that's got to happen. Now, I will say that if you look at an echinoderm, are they a chordate or an invertebrate? They're an invert, right? So, like, what would make them? I guess if you were to look at all of invertebrates, you would put these guys as being like closest to us because they're deuterostomes just like us. In fact, like when you start out and you go like through Porifera, that's the first one. What's the next one after Porifera? Cnidaria. Then after Cnidaria is like Platyhelminthes, and you go all the way down, and at the very end you get to Echinoderm before you jump to chordates. And you save echinoderms for the very end because they share that deuterostome quality with us, basically. Cool. What's next? During mitosis, which of the following normally occurs? This individual had it down to A or B. I think the answer though is going to have to be B. The reason I say that is because what is A really kind of pointing to? Specifically, what part of meiosis? I'd say 1. And in fact, C is about meiosis 1 too. D doesn't really exist. Unreplicated chromosomes like line up at the center. Why would unreplicated chromosomes line up in the center? I don't think there would be any advantage for that at all. This is maternal-paternal chromatids pair. Ooh. Chromatids that's not to happen, like ever. So I think the best answer here is going to be B. Excuse me. A symbiotic relationship. It says that was probably present in the first terrestrial plants, the first land plants, and is still present for the mineral nutrition of present-day angiosperms. I'm thinking right away of bacteria. I don't, again, know if that's necessarily right. What's another like relationship that would exist with plants, though, aside from bacteria? So a fungal relationship. So something bacterial or something fungal. That's kind of what I would be thinking. Insect pollinating flowers, which is totally cool stuff, but the first land plants, were they angiosperms? Angiosperms were like late evolved. Were there insects on land when plants showed up? No. Carnivorous plants feeding on insects. Again, we just already established that insects weren't there. And epiphytes growing on a tree trunk. This says, we were kind of in the zone about how this was first terrestrial plants, right? That means like that there had never been plants on the earth before on the land, right? So how could you have epiphytes growing on trees? Because there should be no trees. Makes sense? And see, and E is actually true. Like epiphytes do grow on a tree trunk, but an epiphyte is like a parasitic plant that grows on another plant. I'd say the best answer here is going to be microrhizae because that's pointing to your fungal, I don't want to say infection, but your fungal association with your root. Yay?
Increased surface area.
Basically, yes. Anytime there's something positive in biology, it's like either coevolution or increased surface area. It's just going to be that. How about 34? Just by show of hands, like how many people are feeling good about 34? Do you want? Why don't y'all try that again? Because I think in almost every class, but maybe 5th period yesterday, I did not get to talking about Hardy-Weinberg equation. Yeah, so 4th period did. Ugh. So Ian, you're gonna go school me in drums. Is that what it's gonna come down to? After this, probably. It's cool. Okay, so looking at a Hardy-Weinberg equilibrium, it says that 9% of the population is homozygous recessive. So that means that 0.09 is going to be what? So what does Q have to be? Now, and I don't know about y'all, and I actually feel like I like math a lot, but for some reason, and probably you might be in this case too, I don't know, but like when it says 0.09 is equal to Q squared, does anyone in here else want to put 0.03? Like I have this, like I feel like this strong need to write that down, and like just remember, like just teach yourself that and get over it, I guess is kind of what I had to do. And then I think, well, 0.3 times 0.3 is going to be 9 for sure. 2 decimal spaces, so 0.09. Like I can get it to work out, but it takes a little time. Definitely, what is the P though after this? It's going to be 0.7, correct? And they want to know, assuming that the population of Hardy-Weinberg equilibrium, which of the following is closest to the frequency of the recessive allele? Oh, we're done. 30%. Yay. What if they said how many people are carriers? Or what's the percentage of carriers? What would you have said? Yeah, about 0.42. 7 times 3, 21 times 2, 42. How many people are homozygous dominant? At what percentage? 49%. 0.7. Yay. It says a characteristic shared by fungi and protists both is both that they are generally, and that's which of the following? Well, let's kind of just talk about each of them. I mean, they're both types of eukaryotes, right? So if I see something about DNA in the nucleus, I'm going for it. Definitely, right? What else is a commonality between them? Haploid at all stages? That's no, wow. Composed of mycelia. That's a fungal thing. Mycelia is like their root system. Progenited, or I'm sorry, not progenited, propagated. Do y'all know what propagated means? To reproduce, to make babies, to continue the lineage on by spores. I kind of recognize that for plants. Fungi too, but probably not protists. Protists, remember, what about protists? If I wanted to find a protist, where would I go? Yeah, bad water, right? As in don't drink it. That type of water. They're single-celled, microscopic almost. Capable of phagocytosis. Again, I would say that's going to be a protist. How do fungi eat, though? This is a good question. How do fungi eat? Yeah, they secrete what's called an exoenzyme. Remember, they digest on the outside of their body cavity. It's extra bodily digestion. I made up that word. That doesn't even exist. But they digest outside of their body. I always like, if people are like, well, how do they do that? I try to like give it an example of you, and this is disgusting, but imagine when you eat, you obviously put it in your like mouth, and then it goes to your tummy, and that's the end of it, right? I mean, it's all personal business from then on. But imagine that, like, to eat, like, let's say you went to McDonald's and you got one of those amazingly unhealthy and not so yummy little double cheeseburgers that they sell with the meat it was from a cow that lived like in the 80s. Imagine it's time for you to break that down. If you were a fungus, what you would do is you would like basically vomit or salivate all over the hamburger. That would break it down, and then you would like put your hand on it and absorb it. That's what a fungus basically does. Make sense? So now you'll never forget that. It's really gross to think of it that way, though. So yeah, definitely don't do that, right? I mean, at McDonald's, because you'll scare away the children. Ronald could never fix that kind of a problem, but that's how a fungus would do it. Which of the following factors is the most effective in changing the allele frequency in a natural population? Changing allele frequency. That's like saying that I don't like tall plants or I don't like short plants, right? how could you control your population, maybe, which is like all the pea plants that you're growing in your backyard, if that's what you were doing. How could you control, and you're like, I hate tall pea plants, and you want to rid of that tall allele. How would you do that?
Is it pretty cool?
I just would kill him. Imagine what would happen. Think about this. If it's a tall pea plant, it's going to be this, or it's going to be this, right? Can I visually inspect if it's a tall pea plant? Yeah? That's what I do. Just karate chop it down, like the Karate Kid. Right? What would be left? Little t, little t. Do you see a big T there at all? Could the big T even be hidden? Like, you know how sometimes you could have, when you have this, you know how you have like the hidden recessive in there? Is there even a dominant hidden in that? He's completely gone, is he not? So I would say selection. Now, what type of selection is that? That's a natural selection. Even though I'm a natural kind of guy. What is it? That's an artificial selection model. Because I'm selecting. Like when you breed dogs or you breed Mendel's little pea plants, that's artificial selection. What's another type of selection? Sexual selection, natural selection. Cool. K capacity, or carrying, or K selected plant species. Remember, we had kind of gone through this the other day. I think we had gone through this a little bit. I know we did. I think it was in 4th period. I had a copy of that packet for chapters like 50 through 54, the ecology chapters. If you didn't get one, we need to get you one from the classroom. Remind me on Monday. Write that down. But the K selected plant species. What about K selection? When we see K, what are we thinking of? Definitely thinking of capacity, right? Says they keep their carrying capacity at a minimum level. I don't like it at all. Why would you want to keep carrying capacity at a minimum? You want to maximize that. That's what carrying capacity does, is it maximizes. They usually reproduce early in life. We're case selected. We don't reproduce early in life, so I don't like that at all. It says they're usually found in climax stages of succession. That's a possibility. Maybe. They are the dominant species in early stages of succession. That's a maybe too. But to be dominant early on, they're probably not going to be dominant. They're a late arriving species typically. They have a short life. Definitely it's not E. I think the best answer is C, and it's not even like a super strong answer of addressing like what a K species is. Secondary tissues of trees. Which of these is going to produce secondary tissues of trees? I think we talked about this in almost every class. I tried to get to this because it was such a strong question. Vascular cambium. Remember, it goes secondary, primary, primary, secondary. Was that not how we established that the pattern was? Was it phloem on the out or on the in? So the phloem was on the outside. The xylem was on the inside. Next is 39. It says, from the list above, which is the most likely chronological order of events from oldest to most recent? This is kind of cool. What's definitely got to be the oldest, though? I would say the oldest thing is going to have to be probably 3. The formation of complex organic compounds. Right, Caleb?
Mm-hmm.
Good job. What should be next after that? I'd say maybe, would 5 be the next one? Would y'all agree? Devin, what do you think?
Yeah.
Okay.
Okay.
So if it's got to go 3, 5, then the answer's got to be C. I did 5 2 though. So 5 2, and I again, this is just me being paranoid. So now you're seeing all my dirty secrets. But does anyone else get really distracted how D and E both start out like the same way, and you want to choose them because that would be, I don't know. It's just me. So let's look at that. Why would 2 not be before the 5? 2 says appearance of chloroplasts, and 5 says development of anaerobic prokaryotes. Okay. Prokaryotes were engulfed. Aerobic prokaryotes were engulfed to become your chloroplasts, right? So I'm not even like the development of anaerobic prokaryotes is like very long ago, not even yet aerobic yet. This is like 2.9 billion years ago. Is like when you're talking about number 5 kind of existing, and chloroplast is like a 2.7 billion years ago bare minimum thing existing. Do you know why I say 2.7? So when oxygen began accumulating. And then realistically, I don't think that number 2 would happen until about closer to about 1.7 billion years ago. In theory, of course. Cyanobacteria don't have chloroplasts, but they became the chloroplasts. So like the 2.7 billion years ago is like when those cyanobacteria that became the chloroplasts showed up, but they at that time were not chloroplasts by definition. They wouldn't have been chloroplasts until they were engulfed sometimes like after the development of a eukaryotic cell, which was like theorized to be like 1.8 billion years ago. How about 40? Question 40. 40. Oh, 40. Let's see. Glass box. Light source. Green light only. This is a lot of plants in this test. Green light only. Let's think about this. So you have a light source that's green light only. What is a plant doing? Like that plant over there. Of course, it's fake, but it's absorbing everything. But what? Green. Yeah, it's absorbing the blues and the reds. Do you remember which absorbs the best, the blue or the red? The blue. Yeah. I always think because blue's like a prettier color than red. I don't know. I just, whatever. So if I shine green light on that, is it going to absorb it? First off, I realize that's not living, so it's not going to absorb anything. But, I mean, it's definitely going to reflect off, like, the green, right? Okay. So no energy would be absorbed. So would you produce any O2? To me, actually, and I kind of dislike this question. I've never seen this before, but I hate how A and D are direct opposites, because typically when you have opposites, like you do with B or E, and you do with A or A or D, to me you're kind of like stuck having to choose one or the other. What is the N2 going to be all about, though? Like, who's responsible for taking N2 and manipulating it? Bacteria. I don't see anything mentioned here about bacteria at all. I don't see any bacteria mentions here at all. So I'm kind of tempted to just avoid the N2 as a discussion, even though I think that it has to be addressed technically. But whatever. Definitely not doing photosynthesis, right? So are you doing what? Respiration only? Respiration only produces CO2, right? So I think we would see maybe an increase in carbon dioxide levels. I think we'd have to see that. Right? Now, naturally, let's just say you had a box that wasn't in, so let's just say we pulled the tomato plant out and we allowed oxygen and carbon dioxide to all freely mix. What should happen to carbon dioxide and oxygen levels over the course of time? In the immediate area, yes. O2 should increase, CO2 should decrease. What about globally, though? If you look at all of everything, CO2 should be, and O2 should be in a static equilibrium, basically, or a dynamic equilibrium, not static. But then you throw that off a little bit when you consider like the change of the seasons. Think about what's happening like during the winter. Are you doing a lot of photosynthesis? So your CO2 level's increasing like steadily throughout the winter, right? Now, wouldn't you not say that like right now is photosynthetic maximum, probably about the time frame that we're in, right? So our CO2 levels are decreasing like staggering levels each day, and we're having more oxygenation each day. We'll maximize the amount of oxygen in about the fall, then we'll like slowly build back up the CO2 only to fix it in the early spring. I think it's kind of fun to think about. 41, it says, the brain most closely associated with muscular coordination. And I like the idea of it being coordination and not just like patternized movement, if you will. So let's remove a couple of bad answers. How about the hypothalamus? Definitely can't be that, right? I think the pineal gland, or they're calling it the pineal body, but either which way, I would eliminate that one. I don't think that has anything to do with it. Let's see. That leaves you with the medulla oblongata. Do you all know what that does? Passed out the brain worksheet the other day. The medulla is all about controlling, like, your O2 intake, like respiration, basic function. So we've gotten rid of these guys. Thalamus versus cerebellum, and I kind of connect thalamus to hypothalamus, realizing that they're probably very closely associated. And honestly, I just know the answer is going to be E, the cerebellum. Like when you have like typing, and like the fact that you can type very quickly, and like you realize like your name. Do you know how like when you're first starting to spell like a new word or a difficult word, you know how you have to sit there and pause and think about it? But when it's something that's like a little bit more natural, it just kind of flows. That's the cerebellum. Like it's a pattern, basically. That's what that part of the brain is working on, developing this massive pattern, working in close association with the cortex. 42. It says the gametophyte generation is therefore haploid. Now I got in trouble last year for the way I teach this, and I'll get in trouble the same way. You probably chose either B or you chose C. I would say majority of you. Definitely I tried to establish the fact that the flower petal was not a gametophyte. The flower structure is a gametophyte, and the reality is, and this is going to cause some issue, but it's the only way I know to teach it and teach it kind of effectively. The answer is actually D, the megaspore. The endosperm is definitely triploid. Now, to me, you'd have a problem choosing B and then not C, because the filament is going to be like all about what? The male or female part? Male part, right? And the cell's all about the female part. But again, if you chose B, you kind of have to choose C a little bit. And the reality is that and now I'll tell you the truth of it, is that like when we say the gametophyte exists, the gametophyte is like really what's only inside the ovary. The gametophyte structure is like not really including your style or your stigma. Your gametophyte is actually just the ovary or just the pollen grain. Sorry. But it's the only way to teach you guys alternation generations, which is more important than this question. Do y'all understand like this idea? You know how like when I draw a flower and I say point to it and that's the gametophyte? It's not it. The flower is diploid like everything else. The style is diploid. The stigma is diploid. Everything's diploid but your ovary and your pollen grain. So it's kind of a beautiful letdown, I think, for some of you now. Like that song by Switchfoot. Number 43. Sequence describes the tissues from the pith outwards to the cork. So they're basically starting you in and going out, looking at the tissue. So definitely xylem is going to be on the inside, correct? Agreed? Agreed.
Did you say on the outside?
No, they want you to start inside and go outside. B is the answer. Yep. Because your secondary is going to be most exterior, I think. Or your secondary is going to be closest to your vascular cambium. Because we had seen that picture the other day. Or we drew that picture the other day. I really think that there's no other way to do that but aside from just like memorize it. Definitely couldn't be E. D is way out of order. They reverse their primary versus their secondary. Another thing to remember is that when you have your vascular cambium, remember that you have like the same type here and here. It's just not, they're not both xylem, they're xylem and phloem. They're both secondary. Fixed action pattern. What was a FAP? Like, yeah, basically the idea that I can repeat myself over and over and over, right? And I know not only is it like to repeat myself, but it's to repeat myself in such a way that like I can guarantee like the fact that this event will happen. It's like it's clockwork almost. Definitely can't be imprinting. Would we say salivation in dogs? Would that be it? Celestial navigation and birds. I think that that's going to be cognitive mapping for E. Echolocation of porpoises. Definitely can't be imprinting. So I think we're like removing A and E off of that for sure. I don't like D because salivation is going to be like they salivate in response to something. They don't like salivation is not a process like where you start you are guaranteed to stop. Salivation is just like it's not even a start stop. It's just a one-time event. It's a one event. It's just to salivate. So maybe I want to get rid of this. Echolocation. I don't really know what that is. Is that like sonar? Does anyone know? I'm kind of guessing it's like a sonar. Is what I'm thinking. That could be for development of maybe a cognitive map or maybe using a landmark. But I still think that multi-steps. FAP, it's got to be like multi-steps where they know to do the steps one after the other. Once you begin step 1, you will finish the 2-step 50. I'd say B. Because echolocation, I only view this as like hitting the sonar button. I don't view it as like being I think it needs to be a process. The first cell of a sporophyte generation, 45 is not too bad. The first cell of a sporophyte is going to be a zygote. Baby zygote, 2N. Punctuated equilibrium. What was that one? Rock and shock, and then all of a sudden, major changes. Like normalcy for forever, basically, and then all of a sudden there's tons of new stuff. So definitely not A. I'd say probably not B either. C, climax communities resulting from ecological succession. Those are like just A through C are a bunch of great words, but they don't make any sense for this question. No, it's definitely going to be D. And it is fossil record proven. It's got proof behind it. The activation of the secondary immune response begins with the activation of which set of stuff? And I would say memory. Definitely not mast cells. Suppressor T cells. Probably like tumor suppressor T cells, if I'd guess. I'd say the best answer is C. Rewards to animals when it behaves correctly, like when we give Tinkerbell her bone. She can go poop in the house. It's definitely a type of conditioning, so yeah, it's got to be an operant type of conditioning. What? Do what? Operant? Who's that? What? Okay, sorry. Like the operant is like based on behavior. Classical's based on like a stimulus. That's the way to do it. I always think of operant as like how they operate. If Tinkerbell operates well, i.e., she doesn't go make number 2 in an inappropriate spot, she doesn't bug me when I'm trying to sleep, then that's a good behavior. So operant's all about behavior, whereas like classical conditioning is going to be like, I ring the little bell and she knows that it's time for her to come inside. She's played enough. Question.
Was 47 the secondary immune response? Was that when you get sick like a second time at the same thing?
Well, no, that's like that. It's not meaning it's a secondary, like the second time I'm infected with this issue, because if they were talking about like the second time that you've encountered this issue, or like the idea that you've gotten a vaccine before, like either one of those would be about antibodies. This is secondary immune response, which is like tier 2 level. The use of basically your memory cells to call forth both B's and T's. You're thinking of the graph in your head, right?
I just thought that I was
It's
with the third.
well, we kind of view it as like maybe a 3-tier system, but really it's more of like a 2-tier system. You have your innate response and you have your active response. You have like the system of like you have the outer and the basic cells, which are your innate, which we called level 1 and 2. and then your level 3 is going to be like your actual real system, which they're calling the secondary immune response.
Okay, so, okay.
And it's not, and my big problem that some people like, if you chose A, I think the A is a perfect answer if you change the word secondary to something along the lines of the second time you are infected with that issue. Like if they weren't saying secondary, if they said like the second time of this, then it would be antibodies, just like the drawing that little graph has. Cool. 49, I have no idea what that question was. I really never was able to see, so I have no idea about it. Let's see. And okay, now the questions like 50 through 53 like are a huge challenge, and
Yeah, like really bad.
this is one thing that you don't really have to like work on, or you don't have to stress about too much, because back in 2008 the College Board was giving out like these little packages of like what they were calling like a required reading material or a suggested reading material, and it really kind of stunk because like it was actually kind of complex reading material, and it all like came to a conclusion during the 2010 like exam, and they asked like this massive essay over like the packet, and they found out that the teachers really weren't getting the message, and I was in the same category of like they wanted the teachers to be passing out these packets to students to have them read, and the scores were so bad that they realized they were going to stop doing that. So I'll go over the answers, but I wouldn't stress it. It says activated by directly binding to GTP. That's going to be a G protein. A G protein only acts if it's like bound by GTP. Think about GTP as activating, which it would do because that's some energy. It activates the G protein. It says it catalyzes the formation of cytosolic secondary messenger molecules. I would say the answer here is A, but it might be best if you answer 53 first and see that 53 is D. The intracellular secondary messenger molecule will always be cyclic AMP. That's not supposed to be CAMP or anything like that. It's cyclic AMP is what it stands for. It's a modified form of ATP minus its two other phosphates, and you cycle it so the molecule like literally looks like a circle. That typically is a secondary messenger that instigates other chemical reactions. Now, the enzyme that makes cyclic AMP exist is going to be the A cyclase enzyme. If I said cyclase, what would you say? Like that's an enzyme for cycling, right? Like that's what you would go for if I said cyclase? Well, so it would make the cyclic AMP. Enhances the transport of lipophilic signals in the blood. So something that's going to like enhance the transport of lipophilic. I think that word could be taken apart. What's lipo mean? Lipid or fat? Philic. So fat liking. Fat liking could be facilitated by what? I'd say you have to have a carrier protein. Cool. These are like my favorite questions, like right through here, because like it becomes matching, and you can start to like really rack up some points. It says this protein requires iron as a cofactor. Definitely, it's going to be hemoglobin. Primary component of skin for 56 is going to be keratin A. Again, this is really nothing more than like a little bit of rote memorization. 56 is A. Yeah, definitely. How about 57? This protein functions at optimally high H plus concentrations. What's that a nice way of saying? What is that a nice way of saying for H plus concentrations? Yeah, acidic conditions or stomach. So we got to be thinking about what? Pepsin. Okay, so B, A, and E are all gone. We still have actin and insulin, and I would definitely put insulin D, not as the answer choice here. I would probably put more likely C, the actin. There's no way insulin's like all about muscle movement. It's all about sugar control. Now they always say that like you can use these more than once, and I've seen that happen like once out of all the AP Bio exams I've seen, but that's only been once out of like at least 50 or 60 of these. So you can, I would say definitely eliminate them, and if it makes absolutely no sense to put your last one or two in the other blank, and it only makes sense to put maybe A in both spots, then do it. Does that make sense? You can always do like the test taker style. Let's say that you didn't know what, you knew the iron had to be used, but you didn't know where it was supposed to be used, then put it like for answer choice of both and like guarantee yourself 1 point. You know that old trick? But like I think here you can probably get something pretty close. It says in 58, part of a chromosome breaks away and then reattaches to the same chromosome in reverse orientation. So you reverse it. I think that would probably best describe an inversion. Part of a chromosome breaks away and attaches to a non-homologous chromosome. So this is like a case where like they're not even like supposed to detach. Non-homologous, I'd say the answer's got to be C, a translocation. That's complete movement. Part of a chromosome, it says, is lacking a centrosome, or centromere. Yeah, centromere. Breaks away and does not reattach to another chromosome. I would say the lacking a centromere, I think, is irrelevant there. Breaks away and does not reattach. What does that mean? Gone forever? It's like deletion. And then tetrads fail to separate correctly during anaphase. This is going to be a non-disjunction if it's there. Yeah, it is. D. When is non-disjunction worse? Meiosis 1 or 2? 1, because they're all screwed up. Meiosis 2, only half of the gametes are screwed up. Would you want non-disjunction to happen? That's certainly like a bad news thing. This is kind of cool. Kind of. Not really. You guys can see what I'm writing, can't you? I recognize that they're all gymnosperms. I'm not saying you have to recognize them as gymnosperms. Definitely the C has to be recognized as gymnosperm, which you might therefore put D as a gymnosperm as well too. And it kind of makes sense. You know you have your mosses, your ferns, your angiosperms. These guys have to collectively be gymnosperms. Now the problem with that is that it's a little difficult to choose for 62. For 62, I couldn't tell you. I mean, I can tell you the answer, and then it's D. I have no way, aside from memorization, that your conifers, like, don't have leaves that are fan-shaped. The fan-shaped leaves is a ginkgo thing. Now, I think definitely the naked seed part, you know, the seed part's going to be right there and to the right. Angiosperms, though, aren't naked seeds because they're encased in a fruit, typically. So you know it's got to be C or D. I think you're down to C or D on that. D is the correct answer, but you're down to C or D. A heart-shaped gametophyte produces swimming sperm. Leaves of sporophytes develop from fiddleheads. Wow. This is so poor of a question. Swimming sperm, though. I was thinking of a fern. They're always going to, like, the fern question, which is also like your pteridophyte group, they are always going to ask you about swimming sperm. That's going to be their question they ask you. The embryo sac with 7 cells and 8 nuclei undergoes double fertilization. Now, double fertilization is something we saw only like in one group of creatures, and that was in the angiosperm. So I think you're definitively on that one. I think 62, honestly, is going to have to be almost a guess between C or D, unless you know the difference between them naturally. I think B you could get for 63 pretty handedly, remembering that swimming sperm is always going to be their firm question. Associated with inactive DNA. So, like, basically the turning off of DNA. How do we turn off DNA? You know, I'm looking at all these answer choices, and the only option that I see that could even make sense is going to have to be A. And methylation does turn off the DNA. Remember, it's acetylation that turns on the DNA, right? Protects RNA from hydrolysis. What do y'all think? I'd say C. You put the poly A tail, the 5' cap. Selectively removes RNA segments, and I see that selectively, like, there's obviously like a reason behind it, and taking out pieces of mRNA. I would say that's going to be what? Yeah, I'd say D, spliceosome. And how about 68? Increases RNA polymerase activity. Yeah, enhancing region. It definitely is not going to be reverse transcription. Okay, so we have these, and I'm not going to read all this stuff up top, but you were doing basically electrophoresis. You have your origin spot, child 1, child 2, child 3, mom, father, and then X. And it says another female X is included for comparison, but no other reason than that. And it says, like, which of these what have you revealed basically from this? And it's just child 1 and child 2 cannot be biological siblings. I would say the fact that they have this common banding pattern means that they could be siblings. The fact that child 1 and the father have the same pattern there, and the mother has the same pattern there, kind of alludes me to that fact. I like how the dad has this piece right there, and these two children have that piece right there. I noticed that if you look at it, though, that there's not a commonality between every which individual. That could be a small problem. Child 1 and child 3 probably look like the mother. 1 and 3. I would say that's definitely not the case. I don't even think child 3 is belonging to the mother. Which actually happens to be question for part C. Yeah, there's no way the mom is the baby of child 3. Because the cuts are nowhere near the same. I'd say C. Y'all agree? E doesn't even make sense. Child 2 and child 3 inherited all their DNA from their father. Like, seriously. That's impossible. The mother's DNA has the same DNA sequence as the father's DNA. Do you understand how D shouldn't never happen? Do you understand what D's saying? D's saying, like, I'm not even going to say what D's saying. D's saying it's not even right. So, we're not going to go there right now, guys. It's all good. Or not. 70 says all the following made the separation of these DNA fragments possible, except the negative charge of the DNA. Is that true? I would say DNA is negatively charged. The percentage of G and C bases. Now, whose principle is that? Do you remember what that was called? Chargaff's principle. Chargaff's rule. But honestly, his rules are relevant here, so I think the answer is going to be B. The porous nature of the gel, I agree. The gel has to allow for the DNA to move through it. It's got to be a little bit of a porous thing. Not that you can see it, because we're very macroscopic. Different fragment sizes. Oh, definitely that had to be right. Smaller moves further. Positive electrode placement. Yeah, you got to put the electrodes the right way, otherwise your DNA is just going to run backwards off the origin. You need that DNA to run this direction, not to run reversal. Runs reversal, you got a problem. It's going to fall right off the edge. Got to go the right direction. So DNA is negative. This negative DNA goes to the positive side, correct? So the DNA is moved. Now, remind ourselves real quick, just in this little drawing. Which DNA moves furthest? The one that is it based on, like, size, right? So is this piece going to be small or big? Super small. And this is going to be a huge piece. Because it didn't move that far at all. Right? 71, it says, which of the following best is the explanation for the fragment pattern for individual X? Oh, this is a little bit nuts because I would say there's a little bit of a similarity right there. Right? It says she only has one living parent. I don't see how B could be important at all. She's homozygous for this particular DNA fragment. I don't think you can tell that. She's not related to any member of the family being tested. Who would you put her related to? I might put her related to... Yeah, I would say right there. So it says she is the mother's child from another marriage. Because there's a similarity between that mom and that daughter, or this kid right here. So, could be D. She's not related. I would say it looks like there is a relation, but honestly, you really can't say E 100% because probably your DNA might cut the same way. She has only one member of this chromosome pair. Or, yeah, we've already established that doesn't make any sense. Okay. It says 72. All of the following are likely steps that would lead to the production of these gel patterns, except you'd have to use a different restriction enzyme. What do different restriction enzymes do? Right. Cut at different spots, you would have different sizes, so you'd have different migrations. Cool? Awesome. Well, I would say let's stop here. We're about 10 minutes away from 11. We're way further ahead than I thought we were ever going to be, which is awesome sauce. It's my new word for the day, awesome sauce. Have you all ever heard that before? Have I said that before?
12.
What?
Now it's like 12.
12? Well, I'm a kid at heart.